Yeah, so my name is Ashley, and this is Andrew Williams and Ian Smith. And so to start, we just wanted to take everybody on a little trip here. Make sure this goes. There it goes. Okay, so we're just going to walk you guys through some of our ecosystems that we actually studied and how diverse they are. So I was looking into the desert ecosystems, and these are highly characterized by being dry and arid and having little to no precipitation throughout the year. And my focus was on the aquatic and environments within these areas, which really rely on all this really little precipitation that they do get.
All right, from the desert, we'll move to the alpine ecosystem. Alpine ecosystems are characterized by being greater than 11,000 feet in elevation, and they take up about 15% of the Earth's surface. They are snowy, windy, cold, and then have really short growing seasons in the summer. And you might recognize some of these spots that I study at. They are just over that way.
Hold on to your seats one more time here. We've moved to where we currently are in Gunnison. Here we live in a high elevation basin that's mostly dominated by sagebrush stepplands, which like many of you, came from the eastern US and noticed there were no trees here in a lot of places. And that's one characteristic of this vast sea of sage, which is a characteristic of a healthy, intact landscape that's home to a lot of rare plants, small mammals, bird species, mainly the Gunnison sage grouse too, one of our favorite birds here, endangered species, listed species, Endangered Species Act. And threats to this environment are, with climate change, it is getting hotter and drier. So drought is an issue, and the increasing encroachment of invasive species like cheatgrass here locally is another issue too. So part of my project works in this environment.
Right, so what do the desert, sagebrush, and alpine have in common? Well, not much, but they all are in the Southwest, which is being heavily impacted by climate change. An estimated 68% of the Western US is still classified as being in an extreme drought, and annual warming that reduces precipitation and snowpack will in turn affect each of the ecosystems for each of our projects and the species that rely on them to survive. And that's all we have for our introduction. So each of us will go through our projects now, and we'll start with Ashley.
All right, so I have been spending the last 2 years looking at landmark-based geometric morphometrics of both bluehead suckers and flannelmouse suckers in the Little Colorado River Basin in Arizona. So the Little Colorado River has been heavily impacted with fragmentation throughout time, mainly with different dams. So we have the C.C. Cragen Dam as well as Lyman Lake, which all reside on the Little Colorado River. The LCR used to be highly perennial, but is now only characterized by being perennial in about 2.5% of the entire system. The rest of it is now intermittent. An example of what this intermittency looks like is shown at Miller Creek right there on the right. And within this system, we have two different species of suckers. So for starters, we have the bluehead suckers. These are not game species, so if you're out fishing, you probably will not be looking to fish for these. But they are highly important and ecologically important, and they're dispersed throughout the Colorado River Basin. And they are not listed federally, but they are considered a sensitive species for a lot of different states in the Colorado River Basin, including Arizona. Within the Little Colorado River itself, they have shown evidence of having a subspecies in the Zuni River, which is called the Zuni bluehead sucker. So, and then also we have our flannelmouth sucker, which are also not federally listed, but they are considered a sensitive species as well. And along with the bluehead, they have also diverged into a morphologically distinct subspecies, which it's not taxonomically named yet, but they do define it as its own subspecies. And it is resides in the upper sections of the LCR as well. So because of these two different diversifications, we were asking, is there any other evidence of morphological variation within the LCR that we can find for blueheads or flannelmouth suckers? And if so, where is this morphological variation occurring? So this is our study area. So right up here, we have our Colorado River, and right here is Lake Powell going into Utah. So within the LCR, we focused on all the different peripheral systems. So we had 7 different systems in different little subbasins. So we focused on Clear Creek, as well as the Winema study site and Silver Creek. So within Clear Creek, there's Barbershop Canyon, Miller Creek, Bear Canyon, and Willow Creek. And then the Winema study site is just what I'll refer to as being the upper LCR and Red Creek. It is important to note that we didn't collect both species from all of these locations. And a lot of our flannelmouth populations were very small. So, whenever it came to sampling, back in 2019 to 2021, the Arizona Game and Fish went through all of our 7 locations using backpack electrofishing, as well as minnow traps or seining. And after they did this, they collected 108 suckers with 20 flannelmouth and 89 blueheads. And at the time of capture, they would take pictures of them, like shown at the right, over here. This would be a lateral view, and this is a ventral view. And the upper is the bluehead sucker, and below is a flannelmouth sucker example. And then after those photos were taken, they took some fin clips, which would be analyzed for DNA later. And then all of these fish were released alive at the site of capture. And now we're going to get into some heavy math. So all of my images were reviewed before I did any of my analysis. So if any of them were blurry or the fish was flopping, I had to exclude those ones. And then from here, I process them in a software called ImageJ. ImageJ allows me to put these different landmarks, 1 through 21, for both views on all of my images. And then from here, all of our measurements were then processed in a R package geomorph. And geomorph actually will allow us to convert all of our different little landmarks into shape variables. This way, we can actually analyze them a little bit easier. So from there, I was able to conduct Procrustes ANOVAs from these Procrustes shape variables. And if I ended up getting any significance from these ANOVAs, then I conducted a pairwise comparison with Bonferroni corrected p-values. That way it accounted for different comparisons that I was doing. And then from here, I also did some principal component analyses. That way I could just visualize all of my different locations to determine if we were getting significant clustering that indicated speciation, as well as doing some deformation grids to show where any of this morphological variation was occurring. And according to the Arizona Game and Fish, we do get different sizes for different ages. So I ended up also running the same analysis for both adult and subadults for both species as well. And then for a total of 13 blueheads, and then only 1 flannelmouth was removed from my study. So we ended up getting about 19 flannelmouth and 76 blueheads. And then it was the same amount for whenever we separated them out by age class, but whenever we looked at the subadult blueheads, we only got like 5. And then we also only got like, I think there was only 3 adult flannelmouth. All the rest were juveniles. So we did get a lot of different native species in our study sites. So we got our 2 species of sucker, as well as the speckled dace and the little Colorado spine dace, which is important because the little Colorado spine dace is a threatened species. But we do have a lot of non-native species throughout. But the main one that we did see was fathead minnow, which is this guy. So whenever we look at the bluehead suckers as a whole, I did find that there was some significance in the ANOVAs. So what I ended up doing was plotting this out on our principal component analysis. And what this is just showing is that there's not really indication of complete speciation. So we would see like major clustering like over here versus like over here if there was like distinct species. And then whenever we look at our deformation grids, the only real thing that we're seeing here is that there's variation in the curvature, as well as within Bear Creek. It's really hard to see on this one, but up towards the right, you can see that's the red dot on Zoom. They are showing some variation going on with the dorsal fin placement, being either forward or back. But and then whenever we look at the size, for the lateral view of bluehead suckers, we are seeing that there's also some variation occurring within the Clear Creek subbasin, within it with the Winema study site. So we're seeing variation with Bear Creek, with Red Creek, as well as with Red Creek and Silver Creek as well. Then whenever we look at the ventral view, we are really noticing that there's a lot of variation occurring within Barbershop Canyon of Clear Creek. And this is between itself, within Clear Creek itself, with Bear Canyon, as well as Willow Creek, and then also with the Winema study site. So that's also very important. Then whenever we look at this by age class, we are noticing that there's also variation occurring within the adult bluehead suckers. So like I said earlier, it's just variation is occurring with the curvature. So as you can see, like with Red Creek, they're a little bit curved up. And then if you look at Willow Creek, it's a little curved down. But I'll show you the actual deformation grids of Bear Creek in a minute. So whenever we look at the Bear Canyon bluehead sucker specifically, what we are noticing is that the dorsal fin placement is either shifted back, which you can see on the upper left one, or it is shifted a little bit forward. So at the very bottom, you can see that it's shifted a little bit forward and over. Yeah, and then we are still seeing the same trends with the other ones. It's just not as prominent as these 2. But also you can see it on that upper right one as well. So for our management implications, this is just showing that both of these species still need to continue in the direction that the range-wide conservation agreement states. So these 2 species are within a 3 species conservation agreement within the Colorado River Basin. So it still needs to stay on track with that because all of our variation isn't showing that there's going to be a different species, but further research into these variations are necessary. So we can start monitoring some of the flow regimes a little bit better to determine if these variations are just a factor of phenotypic plasticity going on from different selective pressures in these locations, as well as analyzing introgression, which is a subpart of this study as well. So all of our fin clips are currently being processed, and we have not got the any of our results back from them. And then we still need to do some more morphological analyses on the larger sample size of a flannelmouth sucker population, just because we did only use like 1 or 2 flannelmouths from each location. So there's not any variation that we could really detect, even if we wanted to. And I really want to just thank Dr. Derek Housen. I don't know if he's on Zoom, but I just want to thank him. He's been a really good support system, as well as Hannah for all of her stats help, because this was really complicated. And so this was really helpful to have her with me, as well as the Arizona Game and Fish for providing the funding for this entire project. as well as my friends and family and my cohort as well.
Great, thank you. We have opportunity for questions. Welcome questions.
So Ashley asked me what got me started looking at these species. So whenever I first got into the program, this whole project was already funded. And my advisor came to me and was like, hey, I got this really awesome project. So I just need some help. So I actually came in with a project already funded, which was fantastic. So I didn't have to go through all the grant issues that a lot of my cohort had to. But that's really the main reason. So he asked me if I was putting all the landmarks on manually. Yes, I was. I spent an entire summer doing that. Yep. So I was asked how I got like the variation that occurred across the grids using my methods. So with Procrustes shape variables, what it does is it superimposes all of your landmarks for the same scale. So then after you do that, you run generalized Procrustes analysis on it, which will give you the entire mean and it'll give you the consensus shape. And so then it'll plot all of your other points to that mean. So then whenever you go in, you can use this function. It's awesome. It's pick and plot. So whenever you look at your plot, you can pick one of them. And what it does is it compares that individual with that consensus shape. So those folds are the change in variation.
Back. Yeah, one last question.
So I was asked if I was able to draw any conclusions on if I was able to find what stressor caused this. No, at the moment we have not. So we're still like looking into why they could be showing this variation. So we are looking into the genetics at the time right now, just to analyze if there's been a lot of introgression going within the populations to see if they're getting some admixture from other populations, or if it's just a result of these shifting flow regimes and that they're just adapting to the different selective pressures.
And we can invite the next one.
So now I would like to introduce Andrea Williams.
The suspense. Cool. So first, I want to ask, raise your hand if you have ever hiked a 14er, a mountain peak above 14,000 feet. Cool. Well, keep your hand up if you've ever hiked Uncompahgre Peak or Red Cloud Peak. Now keep your hand up if you knew that there was an endangered species of butterfly right below these peaks. Nice. More than I thought. All right. Well, so my name's Andrea and my thesis studies the microhabitat requirements of the Uncompahgre fritillary butterfly and climate change implications. And I first want to point out that this guy is an Uncompahgre fritillary butterfly, and I will refer to them as UFB throughout this presentation. So the Uncompahgre fritillary butterfly, or UFB, was discovered in 1978 just below Uncompahgre Peak, and then a few years later below Red Cloud Peak. It was listed as federally endangered in 1991 due to the threats of illegal collection, recreation, grazing, and climate change. And it is found within 11 other habitats now in the San Juan Mountains, meaning it's an endemic species. So their habitat includes northeast facing mesic slopes. Mesic just means wet slopes that are between 3,700 and 4,300 meters above sea level. So just below these 14er peaks. And their habitat also contains their host plant, which is Salix nivalis, or snow willow, which is this pretty little plant right here. It's a dwarf willow, so it's very tiny. And so they use this as their larval food source and egg laying site. But adult UFBs are considered generalist pollinators, meaning they'll frequent various nectar sources during their short 3 to 5 day lifespan as an adult. So for my thesis, I wanted to know first if there are any variation in abundance along transects that are at Red Cloud Peak and Uncompahgre Peak and another site. And then I wanted to see if there are any microhabitat characteristics that might influence UFB abundance at these sites. And lastly, how could we manage the species under climate change conditions, which is a concern for a lot of species out here. So I'll first introduce my sites again. So this is Uncompahgre Peak. It's divided into 3 subcolonies, and it's in that blue square. But it's in the blue square at the top of this map. And then Red Cloud Peak is in the red square at the lower end of this map. That's divided into 2 subcolonies. And lastly, Colony C, which is our confidential site, so it's not on this map, is divided into 2 subcolonies as well. So I have a total of 7 subcolonies. And so the first part of my project was to estimate abundance within the transects that are walked for distance sampling at these subcolonies. Distance sampling is a method of estimating population size where an observer walks along transects and counts the number of UFBs they saw along that transect and the perpendicular distance that UFB was to the transect line. So that's me right here doing distance sampling. And so we have distance sampling data from 2003 to 2021, but for my project, I used it from 2003 to 2020, and I estimated the abundance of UFBs along each transect spanning these colonies, and there's a total of 127 transects. And so with this map right here, this is just one subcolony, and it shows the transect segment indicator on the bottom, and then the estimated abundance on the x-axis or y-axis. And with these estimates, I made a model in R to see which had significantly high or low estimates, and those ones are in yellow on this graph. And then with that information and doing a power analysis in R, I kind of designed my sample sites to include high and low estimated transects. And so this past summer, July of 2021, I began my field measurements, which included taking samples of soil moisture content, slope aspect and elevation, and herbaceous species percent coverage at 700 sample sites between these 7 subcolonies. And I did the herbaceous species coverage with Daubenmeier plots, which are those plots right there. And that's how you just use it to estimate percent coverage of each individual plant species in that plot. And so I had some predictions of what I would see with my data. So I predicted that their host plant or Salix nivalis coverage, as that increased, so would UFB abundance. I also predicted that as soil moisture increased along these transects, so would UFB abundance. And then I predicted that slope and bare ground cover, so the increase in slope and the greater percent bare ground cover would have a negative effect on abundance. And so I took all of that data, took averages of each measured variable, and then modeled it in R a lot of different times to see what had significant effects on UFB abundance. So first I'll go through some of my results. And the first result is that their host plant, Salix nivalis, did show a positive effect on abundance. That's represented in this graph, so that x-axis is the percent cover of Salix nivalis, and the y-axis is the abundance estimate of UFBs. And so I also found that as soil moisture increased, so did abundance, and that agreed with my predictions. And so these results just indicate how important it is that their host plant is in their habitat and how soil moisture might affect these habitats. So soil moisture is key in plant distribution and nutrient availability along alpine ecosystems. And if we're seeing prolonged drought conditions in the United States and in the West especially, that could really impact this soil moisture content. So next, I found that slope actually had a positive effect on abundance, indicated by this graph right here. And that might mean that it creates ideal microhabitats for the UFB, or that it's just manageable by them. So these butterflies fly really low to the ground, and they actually don't move very much during the day. So it could just be a factor that's not affecting them. Next, I found that percent bare ground cover, which is represented by either rocks along the transect or just bare soil, showed a negative effect on abundance, which is in this graph. And that just shows that the lack of vegetation, so anything that causes lack of vegetation, such as grazing or increased competition and species moving up in elevation, could really affect this species. Some more results. So this one was really interesting. I found that as elevation increased along these transects, it was between 3,800 and 4,100 meters, abundance decreased. And so these are high elevation species, but these results kind of show that they might have an upper elevation limit, meaning they can't really move anywhere up in elevation if other species are moving into their habitat due to climate change. So my model of UFB abundance against all of these habitat characteristics also looked at 10 different alpine plant species and their coverage and that effect on UFB abundance. And so these 4 species had a positive effect on UFB abundance, meaning they might be ideal nectar sources, or not really sure why. Maybe they like them, maybe they don't. We didn't monitor behavior of UFBs, but that's really important to consider for future studies. And these showed a negative effect on abundance. So as coverage of these species increased, UFB abundance decreased. So UFBs are considered habitat indicators of alpine ecosystem health. And we know that alpine ecosystems are predicted to be very sensitive to climate change. For instance, the San Juan mountain range had one of the highest rates of warming from 1995 to 2005 in the US. And that's really concerning for alpine obligate species, which 24% are considered to be at risk of extinction due to future climate change. So what can we do moving forward? Well, we can continue population monitoring. This project is actually one of the longest invertebrate monitoring studies in the country, maybe the world, and it needs to continue to provide those survivorship and population estimates of the species. And then we can monitor climate change effects and their effect on all UFB life stages, so at a larval and an adult stage, and then monitor the effects that could impact alpine ecosystems as a whole. Like I said in our introduction, they take up about 15% of the Earth's surface, and that's a lot. So we need to protect these sensitive ecosystems. And then these microhabitat requirements that I found could allow us to find suitable habitat for UFBs that might not have been considered before. And then this overall can give us a better idea of how to manage the species and model their risk of extirpation or extinction, so we can prioritize for long-term conservation of alpine ecosystems in general. I'd like to thank everyone that was involved, my advisor, Kevin Alexander, and all of the field crews that helped me and helped this pass of this project, my family, my friends, my cohort, and the Uncompahgre fritillary butterfly for being a super cool butterfly and for lasting this long. So thank you.
Chance for questions.
Heather asked if they only live on northeast facing slopes. So yes, their slopes are north to northeast to eastern facing slopes. Those just have more sun in the morning, and these butterflies are mostly active during that morning period. That's when we go out and count them for distance sampling. So it's nice and sunny and warm up there early in the morning, and yeah, that's what I think. Yeah. Thank you. So I was asked, how do the butterflies not fly away when you're sampling them? And thank you for bringing that up, because that's an important thing about distance sampling. So when we're walking these transects and counting butterflies, we're actually only counting them when they're in flight. So we're not counting them when they're just resting on the ground. They have to be either flying and you see them, or they have to be flushed. So you're walking and you actually spook one and it flies away. So good point. Yes. So I was asked how I did the soil moisture sampling at the 700 sites. And so I used these soil moisture probes, and I just stuck them in the ground, and they are connected to a data logger. And so that logs the soil moisture content at different intervals. So I would write down the interval to make sure I was getting the right site and the moisture content for that site. And then you just plug it into your computer, into the software, and it just pops up and shows you all the measured samples.
That was the last question.
Cool. Well, next Eli will come up.
Hello, everyone. Yep, once again, my name is Eli Smith, and I'm the sole Master Environmental Management candidate on this panel. My project was officially titled Addressing Climate Resilience in Gunnison County, Transforming the Stewardship Program of High Country Conservation Advocates. High Country Conservation Advocates, I'll refer to them as HCCA from now on. I've been around for 45 years, started out in Crested Butte as a grassroots community-led anti-mining campaign. Their mission is to protect the health and natural beauty of the land, rivers, and wildlife in and around Gunnison County now and for future generations. So over these 45 years, HCCA's had a public lands program, the Red Lady program, which was that mining advocacy wing, and a water quality program. And within the past few years, I started to develop a stewardship program to be more on the proactive side of environmentalism rather than advocacy, which may be a little less accessible to the general public in terms of all the policy that is involved with that. So this was a project to try to get more community engagement within Gunnison County and addressing environmental concerns that are here locally too. Some of those are, like I said, we can look at this through a lens of land and community first. Some of the threats we have going on are, this is in Gunnison County and across the West in general too, but increasing population, increasing tourism, changing climate to one that is hotter and drier. All of this results in increased environmental quality at risk. Most of these projects focus on water attenuation because of the drought risk and trying to address that locally, as well as I mentioned before, in a lot of these project areas is in the sagebrush steppe ecosystem where the Gunnison sage grouse is found, which is a very important species that we are trying to help their population thrive into the future. Right now, there's only, I think, 25 to 3,500 left estimated in this area. The other side I looked at for my project to address was internally at HCCA too. So within the organization, there were 3 full-time staff members who had been working on developing the stewardship program. There wasn't a single person dedicated full-time to this. And that has such a outward-facing, I guess, projects like these. So it's easier to conduct community outreach and get more diverse people involved with these projects. More specifically, the need here is with the federal agencies, state agencies, local governments, and other community stakeholders to identify and prioritize what projects to address exactly on the landscape. Like I said, many of these focus on water attenuation to enhance that conservation and improve wildlife habitat, not just for Gunnison sage grouse, but other elk, mule deer rely on these environments as well. Pointing in more on my project, was looking into with a literature review, talking to people at HCCA with our project partners, what exactly our space can be in that, trying to find our niche. And it turned out organizing the volunteer part of these projects to support the agencies and organizations would be a really great fit for us. These, compared to other volunteer groups that are based here, like Gunnison Trails or Crescent View Mountain Bike Association, do really amazing work with recreation and have a great model of volunteer tours that they offer. And we wanted to do something more on the environmental restoration side to help promote and protect, restore these environments that have been degraded in the past with previous land uses. Part of this going into the stewardship program was to secure more funding. So I applied for a lot of grants this past year. I've been with HCCA since March of last year and applied for 6 grants so far. So that's been a lot of my time. And as well as a lot of internal capacity building with websites, system development, sort of how to track what we're doing, volunteers, all of that. So taking a look at last year, after doing a little bit of a needs assessment and working with the agency partners, we were able to co-host 11 events on the grounds, and that was mostly with the Forest Service, Bureau of Land Management, Colorado Parks and Wildlife, to work on these restoration projects. So up top here, you can see we had 582 volunteer hours given by 107 first-time volunteers and 22 returning from previous seasons. So just a quick show of hands, how many in the crowd have volunteered with HCCA in the past? I've seen a few of you here. All right, very cool. Thank you. Last year, we were able, with these volunteers, to construct 50 rock structures for the Wet Meadows program, and a separate program, the Taylor Park Wetland Restoration, 45 beaver dam analogs. Generously had some lunches donated to us from Firebrand in Gunnison and Germain's as well to feed the volunteers. We planted 300 willows, and had a travel management project in Taylor Park where there were previously a lot of routes identified as being illegal. So we had a lot of volunteers come out and help us close those, construct worm fences, and reseed those to protect sensitive riparian areas found in that area. Yeah, I just want to say it again, all of our projects focus on creating or increasing resilience within these local systems to combat the effects of climate change. It's sort of our guiding principle there. For where we worked this past summer, start up north here in Taylor Park, these two projects were on Forest Service land, and they were that travel management project I mentioned and the Trail Creek Wetland Restoration Project, which is focusing on restoring beaver habitat because there are currently populations above and below the stretch that we worked at. So we created these beaver mimicry structures to build up ponds so that hopefully populations can return and take over the maintenance themselves so that we don't have to be there. The idea in the future. Moving further south here on Flat Top Mountain, there's a large Gunnison sage-grouse population there. And we've been working there for, this project has been going on for a long time now. And it's a great way to get a view of the Gunnison Valley and keep restoring and building these habitats. Further south, these forest strings were all with the Bureau of Land Management, also focusing on the sagebrush environment. Through my time spent as the Environmental Stewardship Fellow with HCCA, I created a volunteer intake system so that we can track volunteers, communicate with them, promote our stewardship work days through HCCA's website, through our newsletters, partners' newsletters. We would write in both newspapers of Gunnison and Crested Butte too, and then report on what we actually did during those work days and share our project successes. We now have a high school intern from Crested Butte Community School who's doing a great job in helping us make connections with potentially youth corps for HCCA, small volunteer corps that is interested in this kind of stuff so that they can get a feeling of what working for an environmental organization or agency would be like. And also was able to partner with a class here at Western, Micah Russell's project management class, pretty much, and they were able to look into financial sustainability of the program. They provided us a literature review of best practices with acquiring funding and looking into an AmeriCorps Vista position and creating a business sponsorship survey to help us look into that for the program specifically. And right now we are developing crew leader positions to help, volunteer crew leaders to help us in the efficiency of these work days so that we can become better at what we do over the years, more established like organizations like Volunteer for Outdoor Colorado or Wildlands Restoration Volunteers who would be part of the model that I've been looking at to kind of have at HCCA. So over the past year, here's a photo first of what these beaver mimicry structures look like on the right there. Work to identify what these needs and opportunities were so that we can have a plan going forward in future seasons. I said, submitted 6 grant applications. So far, 5 of those came in, waiting back to hear on 1 more. So that will also help with the financial sustainability of the program for the next few seasons and just build that base. A lot of this has been happening super fast and in real time. So I've learned a lot on how to be nimble and work with partners. It's been really rewarding in that sense. Next summer, we already have 5 projects ready to go, shovel ready. That'll be at least 10 days. So I'd love to recruit some volunteers here today and just look us up on the website. Come out. It's a lot of fun, as you can see. And continue to strengthen that relationship with Western. It was a really great experience working with that class to have many small projects for HCCA to help us develop our program and for the rest of the organization in general too. And I'm glad to say that we will be welcoming another environmental stewardship fellow from the Indiana program to help us further develop out these projects within the stewardship program. Just a shout out to a few of the partners and funders that we have worked with: the U.S. Forest Service, Colorado Conservation Board, Bureau of Land Management, Colorado Parks and Wildlife, the Town of Crescent Butte, Upper Gunnison River Water Conservation District, National Forest Foundation, the Sustainable and Outdoor Recreation Committee, and Wildland Restoration Volunteers. And in the Town of Crescent Butte, 1% of open space helps fund conservation and restoration projects. Yeah, for my time here in my project, I'd like to acknowledge for funding my position, the SMPS Family Fund and the MEM program for this fellowship that I was in during my time here. At HCCA, this wonderful staff who were great at welcoming into the organization: Brett Henderson, Matt Reed, Julie Nania, and all the board of directors, including Sue Navy, the president of the board right now. My lovely mentor behind the camera, Sally Thode, thank you for everything, being a great mentor. And Micah Russell, the IPLM track director, who was interested in the work that I'm doing and helping connect wetland restoration or within the Indian program right now. And all of the partners were really great. That was just one of my favorite parts of this project, was working with everyone. Somehow the meetings were really efficient and everyone works pretty well together for the most part. It's really inspiring to be in this valley where that happens. So Ashley Hom with the U.S. Forest Service, Andrew B. the BLM, Cheryl Swellick with the Upper Gunnison, Joe Lavarini with the NFF, Max Sauer with the BLM, and MEM, that we partnered with a lot last summer to put on these projects. That is it. Thank you. Some questions now.
Opportunity for questions. Come questions.
Janaea. The question was if I had any repeat volunteers coming out to work days. And yes, last year we had 22 returning. This upcoming year, I've gotten better at creating a sort of a contact list format where I can target those people who have volunteered in the past. And a few of those are going to be volunteer crew leaders. So they're actually stepping up into a higher leadership position because they enjoy the work so much, which is really inspiring. Yeah, happy about that. Yes. Not at this location yet, but they have in the past. I think there are a lot of projects here at Western where they're looking into that and sort of what happens after they are created in the specific area in Taylor Park. But there have been cases where they do come in and take over that human-made structure and they just add on to it and will stay there. Right. The question was about being an advocacy group and doing more on the ground, environmental restoration, and communicating that through grants. So I find a lot of people who are members of HCCA, either they donate money or they're just interested in the work or looking for ways to engage. And so this is a great opportunity for them to come out on a weekend and give back in a way that instills a sense of accomplishment within them and sort of a more personal connection to the landscape that they recreate on, that they live on, all of that. And with the grants, some of those HCCA's applied for in the past, so there's already a relationship built there, and we can report back to the grant funders what we have done in terms of project successes and what we plan on doing moving forward too. And others, it just helps showing examples of what these types of projects are with research listed in there and letters of support from partners as well too.
It's time to invite the panel. for questions for the paper.
Thanks.
And questions, we have here a first question. And one is for Ashley, for our Zoom audience. Do you expect distinct subspeciation to occur in the future within the separate populations?
We do. So because there's been two different events of subspeciation occurring in the LCR itself within both species, we are looking into if there is any further, which we are assuming that there's going to be, just because when you talk to the people in Arizona Game and Fish, they've noticed morphological changes already. So whenever I was working with Mark Grover, who collected my samples and is on my committee, he was explaining to me about how some of these different populations, you can clearly see that there's morphological variations, but he didn't tell me which ones they were. He was hoping I would find it, so, and I did.
Thank you.
So Jess wants us to recount our favorite field moment. I did not spend any time in the field. I spent my time staring at a computer, plotting landmarks, but I think it was my aha moment while I was running my statistical analysis a few times, because there was only one committee member on my committee that has ever run any of this analysis before. And hers is a little bit different. So whenever we were importing our Excel sheets into R, we had to convert it into an array, and it had to be a specific kind of an array. And I spent 3 weeks on Stack Overflow, YouTube, and going back and forth with her that was like, it's not working. I can't even get it imported. And every single time I would try, it would plot this really cool 3-dimensional plot, but I was like, I don't need it in 3D, I need it in 2D. And come to find out, it was a good old comma. So lovely R.
I'll say maybe not my favorite, but my most memorable moment. We were hiking back from a backcountry site, and there was a crew of 4 of us, and we stumbled upon a female and a male moose, and they were just hanging out in the willows very close to the trail that we were on. And so we walk up on them, and the male moose starts doing a very territorial, like, stomping, and you don't want that to happen when you're close to a moose. So we like ran up the slope of the trail, like above the trail, and just walked along the hillside until we didn't see the moose anymore. But yeah, it was very memorable. I won't forget it. And now I know what to do if that happens again.
Labor Day weekend of last year, we had weekend-long volunteer projects, and we had a nitro cold brew coffee sponsor come up on Flat Top Mountain with us, which I had never seen before. So that was really cool to see people buzzing off coffee, just putting rocks together, really enjoying the One Rock Dam sort of puzzle piece in that. So seeing 50 volunteers all down this drainage, all working together, was really cool to see, with Gunnison back down in the backdrop down below.
Okay. Yeah. I was asked if the land management helps plant snow willow, which is the UFB host plant. And to my knowledge, I don't think they do. It is very abundant in the alpine ecosystems. It's the water source for snow snow willow mostly comes from snow, and to my knowledge, they haven't been planting it, but that could be something to consider if other willow species that aren't utilized by the UFB are moving up in elevation, or any other species, or if there's increased grazing or something up there, and we see a decline in snow willow species. So it could be a thing, but yeah, to my knowledge, we don't do that. So we were asked, what do we see within the next 2 years of our projects? So my project, I see doing more studies on nectar source and behavior of UFBs with these plants that I had in my model. And one of the MS in ecology students is actually doing a project starting this summer with a similar route as that. And then I also want to look more into the climate data that we have available for these alpine ecosystems and see if there's any correlation between climate variables and UFB abundance or decline. So that's my plan.
So with my project, it was supposed to be a two-parter, and part of it was supposed to be the morphometrics, and then the other part was the genetic analysis. Just because of COVID and the place that we decided to go through, that has been like jumping through hoops. So we are, I believe, able to finally send off our DNA after 2 years of extracting and playing around with it. And so I'm hoping that within the next year or two, we get at least our DNA back so that we can run all the analysis to see the introgression going on, because we also did a lot more than just those 7 populations. We also got some fish actually from Gunnison as well, so that we can compare some of the hybrids that could be from the Colorado River with the white sucker with some of the ones in the Little Colorado River, which is really cool. Another thing that I think would be really important to look into is just getting like a full idea of the flow regimes that have been going on past, like, I don't know, 70 years in the LCR, just so that we can compare the data and see if that could be leading to the shifts that we see with Bear Creek, just because USGS is very wish-washy on which years that you get data from. So.
Good question. I think there were so many different projects that could come out of further developing like a stewardship program. In terms of community outreach, a lot more to be done with high school engagement. There were so many classes and I think tours that we could offer to project sites to talk about these low-tech restoration methods that are so approachable for people to be involved with and make a difference locally. On the communication side, storytelling, and also always do some work looking at creating a video, a film of these projects coming up in the future as well. Sally. The question was on because projects expanding to different environments. So I think first of all, it would be for whatever our partners' needs are to be. So we provide that boots on the ground labor for them. So if that's prioritized, then yes, I think they would want to go anywhere that's scenic and fun. So I would be into that. But yeah, we work in the sagebrush and then also in Taylor Park currently for the wetland restoration and travel management projects. So the service area is within Gunnison County. So anywhere within that has a restoration need, I think potentially we could go to if we find out all the logistics and coordination with that.
And then we were asked if we see ourselves moving out of our alpine and desert ecosystems to do other work. And yes, I'm not strictly going to work in the alpine. It is really cool though, and it's beautiful, so why not? But there's endangered species everywhere, and I think by working in a very sensitive and remote environment like the alpine, I can learn how other environments might be impacted by factors such as climate change and people. So yeah, and I'm from Florida, and so alpine is very different from where I grew up. So I kind of have a mixture of ecosystems I'm interested in, and I'm not limited to just one.
Yeah, I'm kind of the same. So as much as I love my desert ecosystem that I've been working with, I have a soft spot for the mountains. And I currently got a position up in Idaho, so I will be looking at fish population dynamics up there. So I'm still going to stick with some fish for now, and then possibly move to vertebrates or terrestrial, and get out of the fish a little bit. I don't want to be strictly one thing. I'm like Andrea.
Congratulations to this panel, Andrea, Ashley, and Ellie. And if you invite the following.
Thank you all, and thank you for such a riveting panel. I learned so much. Again, I really, really appreciate the chance to learn from all of you. And I feel like if we could just sit this panel in a room for a little while, they could come up with all sorts of creative ways to engage people in species conservation and increase our understanding of our ecosystems near and far. So thank you for that. And it's now my great honor to present these folks with their cool pens, but symbolic of their degrees and commemorate the hard work that they've done to earn their master's degrees. So please join me in congratulating Ashley Hurst, a Master of Science in Ecology. Andrea Williams, Masters of Science in Ecology. And Eli Smith, Master of Environmental Management. One more congratulations to this amazing panel.
Now my pleasure to introduce some of my favorite people in the Clark School. I really mean that for their projects. Panel, place-based management of wild western forests and forests. Emma McClure, Kristen Hinkie, Abe Prophet, and Alex Stack.